First, I want to say it's an honor to be on this presentation, and I want to thank Global Bio Investment and all of the other support issues, and also emphasize that the involvement of the law group is really an important part of what the message I'm going to be sharing today. My name is Alan Liss. I have background in microbiology and immunology. I've been fortunate to have experiences in both college, university teaching, independent and large manufacturing companies, and retired from the US FDA. When I did retire, one of the first things I wanted to do is to visit Taiwan. And there I met so many great people and so many great pieces of the Taiwanese government infrastructure, the FDA and Ministry of Health, all with the understanding that even though Taiwan was small, it could really contribute greatly to global public health issues. And I hope that this is the beginning of the next step in Taiwan's influence on health, not only within the Asian sphere, but also globally. Next. Okay. So as I said, you don't. I have in this slide deck things that hopefully you can take home with you later. I won't go through everything in detail, excuse me, but I've been fortunate to have a varied background. And in that varied background, I think there's been one consistent thought, and that is to continue to move forward. And I think this is still the theme that as I work with as a consultant, and making sure that we all have the opportunity to try new things, all have the opportunity to make mistakes, but more importantly, we respect the concept of learning from all of our mistakes and things that we do. And certainly those aspects fit into the evolution and the maturation of cell and gene therapy. Next. So we all know that when you develop not only something at the research level, but your intention should be, I would hope, is that some of your great experiments will become great products. And this is a long process that takes lots of time and lots of money, but it's something that is well worthwhile. And hopefully no one should be prohibited from doing research and from doing experiments for publications. But I also hope some in the audience understand that some of their work may actually go into humans in the future and be very helpful for improving the life of, again, not only us, but everyone else in the world. Next. So both Ira and I have been honored by being part of the United States Food and Drug Administration. And it's a large organization with lots of divisions in it, but there's one common theme, and that is that FDA promotes innovation as well as promoting safety and hopefully efficacy for the population that may be taking these products. So this is something that is quite large and complex, and as you may have been hearing in the news, has been recently affected by people from our new government that don't necessarily understand what the mission of the Food and Drug Administration, both in the US and around the world. Next, please. Next. Okay. So this is not a meeting of the leadership of FDA, but I do want to warn you that strange things have happened already and will continue to happen. The bottom has not been hit. So the important thing for you all is to focus not only on your research, but have another eye open for what's happening in the US and in the rest of the world. And certainly Ira and I offer you as to be conduits for some of this information. But we are in a situation probably unlike any in our history of strange things happening that will have significant effects on global public health. But with that in mind, let's move on to discuss our topic for today. Next, please. Next. So cell and gene therapy sounds like a simple phrase, but it really involves a great deal of different kind of products. There are cell therapies, which, as you can imagine, involve using cellular therapies to change and affect some type of defect. Next slide, please. Next. And whatever we look in the future of products as far as the advanced development manufacturing type of therapies, certainly cell and gene therapies are high up in the list of things that's going to make and break the future. Next. Any statistic you look at shows that there's incredible growth. And this is really an amazing situation, since a lot of this growth is not occurring from your larger companies, but in fact is either occurring from hospital-based research or small startup companies. Next, please. Next. So there are probably three buckets in which most of the cell and gene therapies can be placed into it. In vivo therapies, ex vivo therapies, and unmodified human parts put into the same human again. So in this short time that we have, it's going to be impossible for us to talk about all of these. So I'm going to try to share with you a factor that is common to the in vivo and ex vivo, and has been particularly important in looking at products that have failed to get approval as we move through the regulatory process. Next, please. So one of the things that where the Law Institute is involved in the US as well is that the regulations and guidances from the US FDA are all based on laws that are in our various books. And we have different buckets of those types of issues. And I'm going to again focus on those that are considered drugs or biologics. The same surgery where you have an organ or a part of you that is replaced by the same organ that was there originally, or synthetic processes, for example, knee replacements. We won't talk about those today, but I want to just focus again on the center of this, which is most of the probably cutting edge research that's going on. Next, please. The important thing is that all of the basic information foundation of any type of regulatory science is going to be based on the fact that the FDA is involved. And what the FDA is looking at is that there is control, and this control should aid in the recognition of safety and maybe even in the effectiveness of particular products. So all of this is then supported by actual laws in the Public Health Service Act. And what this is important is the fact that many times people forget that the properties of good manufacturing processes, the GMPs, have to apply to these products, even though they may be born in a lab situation that seems to be quite controlled. But as I'll be talking about continuing in the next few slides, these are things that have to be regulated by the US FDA as if it were a small molecule or a recognized large molecule. Next slide, please. Next, there we go. So I'm sorry, could you go back one? Okay, so the concept of a human cell therapy and the manufacturing process that under which it goes through is something that is perhaps one of the stumbling blocks for many of the new products that want to go from research into a human situation. And many times the flaw is at the very beginning, that the manufacturing facility is atypical. It's not what you would expect for a sterile product. So it's never too early, even in your research aims, to be thinking about practices that have to be used under a controlled environment, so that were you actually lucky enough that something actually worked and made it into a human clinical trial, that you would be available to be inspected. And in my retirement and consulting days, that's probably the number one thing that I find I'm doing is doing pre-inspection audits. And you see a lot of very good research being done in some very strange places. And unfortunately, that ends up in the research never making it to that next level of human use. Next slide, please. So this is a new field. This is not only a new field for us as researchers, but it's certainly a new field for the regulators. And this is something which I think Taiwan could make a really important contribution to. And that is that innovation from researchers usually comes on much faster than innovation from the regulators. And I hope that you can understand that the partnership between regulators, the Ministry of Health, and researchers should be one of a mutual sharing of concepts, so that you can generate some type of consistency, robustness, and control. Probably the most important thing that happened in the US was in the early days of biotechnology was the involvement with the US FDA on not financially supporting any particular companies, but participating in learning about all of this new technology and understanding and sharing how sometimes cutting edge research could not necessarily be an approvable regulatory science type of experiment. So next slide, please. When you're regulating the cell and gene therapies, you have to understand that in the US, every regulation and every audit, for example, comment is based on a particular section in a law. What this means, though, is that the inspectors and the reviewers have to be up on the science that they're looking at, because most of the laws and regulations are designed for small molecules, which is complex but simple chemistry compared to the more complex chemistry of biologics. And now when we're talking about cell and gene therapy, we probably have a even more complex situation. So one of your missions as researchers and new product producers is to be able to educate regulators about what you're doing and how and why that is controllable and safe. Next, please. Next. So this all comes down to really one most important question from the CMC side, and Ira will talk about the other end in the clinical. But that is, what really is your product? And many times I hear that people are expecting the regulators, including the US FDA, to define what their product should be. But you need to understand that, particularly when you're on the edge of a new science, as we are here, you have to be the one who defines what your product is and should be. So this can be problematic, but it generally is something that you will learn to appreciate as you make and use more of your product in your various animal models and even in your clinical trials. Next slide, please. So a concept of recognizing new science is based upon perhaps the most important aspect of developing a new product, and that is safety. And safety in a clinical situation often is a risk-benefit situation analysis. So part of your thinking should not be simply that it's either really great science, but you should also be thinking of what you're trying to do and how you produce your particular therapy. Does it add any risk to the patient? And this is often a situation of balancing, again, the risks of someone not having a therapy versus the risk of a new therapy. And again, there are no simple answers. There are no published papers telling you exactly what the risk should be. But as regulatory scientists, that is someone who's trying to develop a product, not simply some interesting research. This should be part of your consideration. Is am I helping the particular therapeutic endpoint, but am I hurting the patient as a side effect of what I'm creating? Next, please. Now, viral vectors for gene therapy, and I'm sure there's no way you can read the slide, but when you get the copies, you can look at this. Viral vectors for gene therapy, probably one of the most quantitatively active areas of cell and gene therapy, particularly gene therapy. And there are a lot of different vectors that are used in this process. Next slide, please. And it's important that when you develop and select a vector, that you really understand exactly, is that the perfect vector for what you want, or is that happens to be a convenient vector at the time? And there are cases where particular vectors simply cannot be either used to increase the scale of your project, or they can add some type of risk to that. So non-clinical testing is incredibly important when you're developing a new platform, a new process, a new product. And the guidances for this are pretty much developed by regulators for products that are more or less complex. So in many cases, you're going to have to develop your own particular model and then be able to explain why that's purposeful for developing a safety profile for your product. Again, in regulatory science, the regulators don't necessarily have all of the answers. Sometimes they don't have any of the answers. But as you, as an owner and a sponsor of your new product, you need to be thinking not only of the science, but also the ramifications of this potentially being used in humans. And will it add any safety factors while it's trying to help? Next slide, please. So when you know your product, one of the things that FDA has been trying to do is come out with a variety of documents. We can call them guidances, we can call them directives, to help define what they're actually looking for. Not what your answer should be, but what they're looking for. But even in those attempts, it can make the issue of defining a product quite complicated. For example, there have been discussion in some draft guidances about release testing of your product. So in most simple small molecules or even well-defined complex biologics, a release test is pretty reasonable based on the chemistry and science of the manufacturing process. However, in the case of some therapies, particularly cell and gene therapies, the time that you would have to take to develop and actually execute these tests on your putative product may be too excessive, and or it may be, in fact, in many cases, the product that you're making is in such small quantity that you can't do too much testing, or you'll have nothing that ever goes into the patients. So you can make decisions about how to define what release testing is, what in-process testing is, but you need to justify that based on the science of what you're working with. Again, don't expect the agency to have all the answers. You have the obligation of educating the regulators based on what you know and the experiments that you've done, that you are the right path. Next, please. So if I had to pick one issue of failure that I've seen in various cell and gene therapy products, that has been the product of the lack of stability. Next, please. So stability testing is a really critical aspect of making a product that is well controlled and safe enough to go into humans. And stability testing, by its very nature, is based on you're defining and establishing that the tests that you're doing are critical for that product to be safe and effective. So if you don't know your product, it's going to be really difficult to develop stability testing because you'll probably do what we see on the next slide, please, is use lots of different tests. And this is one example of a variety of tests that people have used for their therapies. And generally, the less you know about the product, the more likely you are to just test the heck out of it. But that's not really the most effective regulatory science way to do this. So the more you know about your product, the more well-defined and consistently you make your product, the easier it's going to be for you to demonstrate that it is stable based on the knowledge of your process. Next product, please. So I can't emphasize too much how the early studies that you do will help you define your product, will help you define the stability. So these can be animal models. These can be cell culture models. Whatever, if it's possible to get some firm understanding of what your product should look like, and particularly when it's effective, it's going to help you move into the next level of working with humans. So if there are animal models, if there are cell culture models, these will all be a great value. And again, there is no one animal model or one cell culture model that's going to be effective for everybody. You have to justify it based on what you're trying to do, your clinical endpoint and your manufacturing process. Next, please. So one of the side effects of why are we asking for so much data can actually be a very interesting part of our science, and particularly in the business end of it. Next. So in many cases, you can use orphan drug exclusivities as something Ira will talk about in his aspect. And that is simply looking at a small, unique population gives you an edge, both research-wise, but particularly in regards to the business end of it, of actually distributing and selling your product. Next. And one of the aspects of orphan designation is determining the sameness of your product or the differentness of your product compared to another product. And this isn't possible if you don't understand and don't have a well-defined process and a well-defined product. And why is this important? Why are we worried about well-defined and sameness and knowing your product? Well, we have a kind of a sister field. Next slide, please. Next, we can skip this one. So, oh, sorry, go back. I wanted to just, there you go, that's good. So the sister field is in the area of monoclonal antibodies, and particularly even antibody drug conjugates, which are very popular as well. And what happens is that as part of the business aspects is the competition to make what we could crudely call a generic. And the more you know about your product and the more details you can about your product, the more likely you can block other companies, other researchers from developing a similar product. So this isn't simply a generation of lots of data for no particular reason, but this can be a very strong business aspect, particularly if you're a startup company looking for funding and you're trying to have uniqueness. So knowing your product can help in both simple scientific and regulatory science ways, but it can be also a really nice leverage for bringing in money to help your business grow. Next, please. So in summary, wherever, in whatever part of the cell and gene therapy ecosystem you're dealing with, you have to understand that part of any drug going into humans has to be its safety profile. And you have to understand that part of the safety profile is establishing and making your product, even at the early stages, under situations that could pass an inspection. And it's important as well that you have a well-defined process and have your assays well-defined and qualified so that when you get the answers to defining your product, it can be accepted and established from the regulator. Next, please. So if I had to summarize all of this for early stage interactions, for making actually the cell and gene therapy products. Communication with the FDA or your local regulatory global agency is quite critical. It's important not only to find out what they want from you, but it's also critical for you to explain to them anything that's really new so that they understand and can accept your new whatever when it comes to them as a reviewable document. Your manufacturing considerations, even at early stages, should be part of what you're doing. Nothing is more frustrating than having something that can be done in a particular laboratory at a particular small scale, but can't be moved to another laboratory, another hospital, or scale increased. And this manufacturing should be in an environment that is controllable, and we can use the term GMP about that. So good manufacturing, good practice. As much non-clinical data that you have can be very, very critical. And this can develop your safety profile and make review of your product before it goes into humans as it can just be a priceless aid. And finally, as we'll hear more about from Ira, obviously the clinical target that you're looking at should be a motivator for you. And yes, it's important to do a lot of good research, but some of you, and I hope many of you, are also considering that your research will be used to cure unmet medical needs that exist today. And that should be one of the motivations for all of us to be working hard in this new area. So with that note, I want to pass the ball to Ira, who is I've been honored to be one of his colleagues, and he'll tell you his story.